both classes today. Hardly anybody here. Glad you all can make it. Is it the weather? Rainy, cold, snowy. Kind of a good day to put the blankets over your head and just keep sleeping. Okay. So I will be posting more homework assignments, gearing you up for the final exam content. And so I'll be trying to remember to send out announcements as soon as I do that to give you a heads up. And then, oh, what was the other announcement? There's something else. Oh, I'm still working on grading the essay portion of the exam. I'm hoping I can get to that through the weekend, and so by next week I should have that done. So I'll send out an announcement when I get those grades posted. Okay. All right. So we are looking at soil system. We've looked at this, how we have basically from the pH 5.5 to 6.5 range, we have minerals more available to the plant, which means they're more soluble and readily taken up from the soil water solution. We were thinking about fungi being more prevalent at that pH as well. Fungi do like slightly acidic conditions, whether you grow them on an agar plate. When I work with them in the lab, I always have to provide them with slightly acidic conditions. And here we looked at this, how you see the mycorrhizal fungi, all the hyphae or the mycelium growing off the roots that are thicker and bigger in diameter. But you have these nice fine hyphal strands that will grow out into the soil. And then I think we left off where we were looking at this idea that not only do you have the fungal hyphae there growing off the soil, but it seems like they have their own little system of bacteria that come along with them, kind of skate along the surface of their hyphae in a thin film of water. And it's the action of both the fungi and the bacteria that are in this particular paper. They were looking at phosphorus in particular, because phosphorus is definitely a trickier element for plants to get access to. Probably more so than even nitrogen in some regards. And so I think we left off here looking at how in that video clip the bacteria were skating along quickly actually along the hyphae, using it as like a highway system. And so this idea that you don't have the plants living in isolation certainly holds true. Even in a hydroponic system, you still have microbes coexisting with the plant. In a basic soil system, you definitely have mycorrhizal fungi capitalizing on their ability to get into smaller nooks and crannies of the soil spaces so that they can physically get to things that the plant roots cannot. And I think we've covered this already, but I wanted to remind you that in particular, I've bolded the P because phosphorus in particular is greatly, the uptake of that is greatly enhanced if you have the mycorrhizal fungi present. Also nitrogen and potassium though too. And those are the three big players. When you look at any fertilizers that they sell commercially, it's always NPK that is being sold in that package typically. And so mycorrhizal fungi are just really good at getting access to phosphorus. And so the fungi have the ability to like solubilize phosphorus. So if phosphorus is attached to the soil particle, the plant can't get to it directly. If it is complex with another set of molecules and precipitates out of that water solution, the plant can't get access to the phosphorus in those conditions either. But the fungi can. Mycorrhizal fungi have all kinds of funky enzymes and tricks up their sleeve to get access to these minerals that the plant can't directly grab onto. And also, too, of course, mycorrhizal fungi are excellent at helping the plant get access to water in the soil system in general. Okay, so don't forget about the mycorrhizal fungi, also the beneficial bacteria hanging out with the plant in the soil system. But let's look at some like what do they really need besides NPK? What else are we thinking about? And so they break them down into macro versus micronutrients. And that's simply looking at how much do they need of these different minerals. So macronutrients just means that they're required in larger amounts. And the reason they're required in larger amounts is they're usually involved in some kind of structure inside the plant body. And so it takes a lot in order to build that portion of the plant body. Micronutrients, on the other hand, are required in small amounts, but that does not mean that they're not as important. They're just as important as the macronutrients. They're just required in smaller amounts. And the reason for that is they tend to serve more of a catalytic role in physiological pathways, regulatory role. And so you don't need a lot, but you certainly need them. Otherwise, their physiology just falls apart. And so macro versus micro. And then that last category, so they've studied, all of these different mineral nutrients, and they find that this list, which I'd like you to memorize, please. NPK, magnesium, calcium, and iron. Please know that those are the ones that it seems for most plants, especially in commercial production, most plants, if you don't have enough of these, it's definitely going to impact the growth of that plant. So NPK, and then magnesium, calcium, and iron. So the only one on that list that is a micronutrient is iron, Fe, iron. We'll talk a little bit more about all of these though. Okay, macro versus micro. Here you're looking at a list of table showing a list with they're putting it into perspective of molybdenum, because molybdenum is typically the lowest required in terms of amount. And then they put everything into perspective of that. And so we're focusing on the lower 2/3 of this table because plants can get carbon, hydrogen, and oxygen from water or from carbon dioxide. So we're not really thinking about those. We're thinking about what's going on in the soil system. So the macro and the micronutrients. And definitely NPK. That's why most fertilizers have NPK in that one container of fertilizer. They need those in very large amounts. And then the other ones that tend to show up as pretty high need is calcium, magnesium, and sulfur. And then usually micronutrients, if you purchase micronutrients, they usually come and they're often labeled as trace elements. And so that's another common way to refer to them. Another thing that I don't have time to get into in huge detail, but you need to be aware that minerals can interfere with each other or interact with each other, and that can cause problems. So some are synergistic in the sense of they work well together, and it really, those minerals really enhance each other's uptake, for example. Some, however, are antagonistic. So, for example, in terms of synergistic, calcium can help potassium uptake. So that's kind of a nice synergistic thing. But then calcium and magnesium don't work well together. They don't play well together. All right. So they are antagonistic to each other in terms of uptake. And then if you have too much of one, that can often cause a deficiency of the other. And so it's complicated. And so knowing a little bit about this, for those of you who've taken soils, I don't know, does Dr. VG get into any of this idea at all? It's probably, if you've got so much other stuff to talk about, it probably can't make it onto the schedule, but it's complicated. And so, when I take soil samples, I always send it off. I have a couple of reputable companies and actually some universities that will test soil and plant tissues and tell you exactly what's in there. And then they can give you feedback. They usually have technicians on staff that will be able to answer your question so that you can get a better feel for some of the details of what might be going on in your soil, what's good about it, what's not so good about it, and how to improve it. So some people just think, oh, more is better. I'll just dump more fertilizer into that system. And yeah, you got to be careful about that because sometimes you can create problems that you just didn't envision in terms of antagonistic mineral interactions. So just be careful about that. It's always good to consult with an expert. Okay, So here, this is from the book. So you're looking at examples of like page. So plant needs NPK, but why? So this table is looking at what does that do for the plant. So I would like you to familiarize yourself with these ideas. Okay, just within the table. This is kind of a synopsis. There's more to it. Like nitrogen can be used for more than what's just listed, but these are good examples. And so please be familiar. So for example, on the final exam, I may describe what this element does for the plant, and I'll give you enough detail that you should be able to pick from a list of 4 or so which one that is. Okay. And so you can see, there's a lots of different reasons why these minerals are needed. Makes sense when you think about a multicellular, complicated organism like a plant. And then the micronutrients too, you're looking at a list here. Please be familiar with these as well, especially we'll kind of focus in on iron a little bit. And then this table is not from the book, but I'd like you to know this table too, which is looking at once the plant takes up the mineral, then we think about, okay, is that mineral going to end up being mobile through the plant body or immobile? What they mean by that is if the plant takes up nitrogen, for example, you can see it made the list as mobile. So then the plant takes up nitrogen, puts it into its leaf, let's say, and then the plant keeps growing, and so new leaves start to grow. And then the plant experiences nitrogen deficiency in the soil. So, but these new leaves need that nitrogen. And so because nitrogen is mobile, what happens is the older leaves sacrifice their nitrogen and they'll send it to the younger leaves and allow the younger leaves to get off to a good start. And so what you tend to see then is that the fancy word is it can be translocated or moved from the older leaves to the younger leaves. And so then the older leaves start to show deficiencies first. And I'll talk about what do some of these deficiencies look like. And so you can see here the list: nitrogen, potassium, phosphorus, so the NPK, those are mobile, those magnesium. But there's others on the list here. I've highlighted zinc in yellow because that's debatable. I've seen many different tables of what's mobile and immobile, and I can't get a clear bead on zinc. But I'm wondering if it's more species specific, perhaps, that sometimes it's mobile and sometimes it's immobile. So just be aware of zinc. There's a little bit of a debate on that one. If the mineral gets taken up into the plant tissue and put where it goes right away, then if it's immobile, that means it can't get mobilized in the plant body. So iron cannot be mobilized. Once the plant puts iron wherever it gets put, then it initially is brought in. If a younger leaf is iron deficient, too bad. That younger leaf will not get any iron remobilized or translocated to the younger growth. And so then the young tissue starts to look, has deficiencies. Okay. So this is actually something that you can use in your everyday life, even if just with houseplants. If you are wondering if you have a mineral deficiency and you're trying to figure out what that might be, you should look for these patterns of where is the deficiency symptom showing up. And based on that, you can start to narrow it down. And I'll talk to you a little bit about how to read your plant in that regard. So for example, here are 2 pictures. One is showing a mobile nitrogen deficiency. So this is a cornfield, and you can see that the older leaves, remember now older leaves are at the bottom of the stem on any plant. And so the older leaves are starting to turn yellow and look deficient compared to the new leaves, which are still quite green. Iron, on the other hand, the newer growth up top, you can definitely see something's going on there that's not so good. You got some definite yellowing going on there, but the older leaves are just fine. So this is an immobile pattern of deficiency. Okay, so clearly very, very different parts of the plant body, depending on if it's mobile versus immobile. So let's look at some of these deficiencies. Here's some images, and you're looking at basically when you say, oh, that plant looks chlorotic, or look at the chlorosis on that plant, you're describing a yellow color to the tissue, right? And then depending on how that yellow shows up, so the far left one, this is actually a nitrogen deficiency. That is called general chlorosis, where the entire leaf has gone yellow. So general chlorosis. Now the middle pictures are showing mottled chlorosis, where you have speckles or dots of yellow interspersed with green. And that can be a key thing to pick up on, these different patterns. And then the last far right picture is interveinal. So some deficiencies, the veins still pop green, but the rest of the tissue has gone chlorotic or yellow. And then when things get really bad, you start talking about necrosis. That's when the tissue goes from yellow to brown or black. So brown or black means dead, dead, dead. There's no coming back from that. And so you can have general necrosis, mottled necrosis. Yeah, I don't know if you can get intervenal necrosis. I don't know if I've ever seen that, but okay. So these are the patterns you should train your eye to, even with your houseplants, because that can tell you a lot in terms of what type of deficiency. Now, having said that, there are deficiencies that they're shared. So sometimes two different minerals will display very similar symptoms. So, for example, that especially general chlorosis, you can see that happening with several different deficiencies. So you can't always precisely say, oh, it's a blank deficiency. You have to use other clues than just looking at the plant in order to really get it narrowed down. So what I'd like to do is go through a handful of the minerals, not every single one, but some of the more important ones, and show you a little bit about that mineral, why the plant's using it, and is it mobile or immobile, what do deficiency symptoms look like. So here you have nitrogen again, the corn showing the deficiency symptom of chlorosis in the bottom leaf. And so when you look at nitrogen, we have a ton of it in our atmosphere. We're sucking it in right now. 80% of our atmosphere is dinitrogen. But remember, plants cannot directly access that. Only bacteria can. And then you have plenty of nitrogen in organic tissue, whether it's plants or creepy crawly organisms in the soil or all the different animals, whatever. And then there's inorganic forms of nitrogen. And so for the plants, they care about nitrate, NO3 minus, and they care about ammonium, NH4 plus. Okay, those are really the two forms that can be absorbed by the plant. The plant cannot grab dinitrogen from the atmosphere. It has to have help with associated bacteria. So it's really nitrate and ammonium. So you need to know those two forms. Those are really important. If they're going to use nitrogen, AA, that abbreviation is amino acids. So they're using it to build amino acids, which have plenty of nitrogen as part of the chemical makeup. Certainly proteins, Na is nucleic acids, DNA, RNA have plenty of nitrogen. Hormones in the plant body are often have nitrogen in the structure. And then CHL is chlorophyll. Okay, so chlorophyll also has nitrogen. So you can see these are heavy hitters. I mean, all that in that list, plants have a lot of all of that. And so it makes sense that nitrogen is needed high. Yes. They can work with nitrite in the sense of they have physiological pathways, but they usually convert it to nitrates, which then usually get converted to ammonium pretty quickly. Yeah, they usually take that and run. And then like, I don't even list on here. Remember too that that's supposed to be an N. NH3 ammonia is interconvertible with NH4 plus, but I don't have that listed out here because ammonia, NH3, is poisonous to all of us, including plants. And so they can work with them though. If they get NH3, they can quickly convert it to NH4+, which is less serious of an issue. And so I think that's what they actually put in like smelling salts when you faint, and they crack that thing and put it under your nose and you inhale it and you wake up out of a dead faint. I think it's ammonia because it's so toxic to us. It wakes us out of a dead faint to get the heck away from it. And so pretty serious chemical. But anyways, nitrogen needed in very, very high levels. Very mobile, so that's why the older bees show chlorosis first, and it's usually general chlorosis. If you keep the plant growing and they stay deficient, their overall biomass is much lower. They just don't grow very big. It takes them a lot longer to form flowers. The other thing to key in on, which is sometimes very, very obvious and other times not, is this anthocyanin accumulation. This is a pigment. It's purple. It's really pretty when you look at it. It's beautiful purple pigment. And you see it showing up on the stems of the plant, the stem of the leaf. So the petiole is the little leaf stem. And then you have to turn the leaf over and look at the underside. Sometimes then it's purple there as well. Now, this isn't unique to nitrogen because phosphorus deficiencies do this as well. So just because you see anthocyanin, that purple pigment showing up, doesn't mean it's nitrogen. Okay, but it narrows it down. It's nitrogen or phosphorus. Those are the two classic deficiencies that show anthocyanin. But not every plant will do that, only some. Okay, so it's mobile. So, nitrogen fertilization is very, very common. You add fertilizers to your fields to grow plants or into your potted plant in your home. So it's actually, when you look at how they do it, this little schematic is looking at how they make ammonia type fertilizer, which again, the plant can quickly take that and make ammonium from it. They're showing here that it's very high pressure in order to do this. Lots and lots of pressure. That's incredible pressure, incredible temperatures. This is very, very energy intensive. You have to pump in methane gas to get it to happen and take in dinitrogen from the atmosphere. It's actually kind of similar in terms of the ultimate start and end, because you start with dinitrogen, N2, and you end up with ammonia. And that's exactly what the bacteria do that hang out with plants, fix nitrogen. When humans do it, they use a whole lot of pressure and temperature, lots of energy to get this to happen. So the bacteria are way better at it than we are. And so making ammonia fertilizer is energy intensive. It is not easy to do. And technically the name of this is called the Haber-Bosch process. You might have heard that term. After the two scientists who came up with it. I believe this was generated during World War I or II. They figured out this process. And so also it generates a fair amount of carbon dioxide, which for global warming issues is not a big deal. And so the fact that bacteria can essentially do the same thing, but not just at regular normal conditions is pretty amazing. We have to resort to really extreme conditions to get this to happen. So when bacteria do it, they get fed carbohydrates from the plant. So the plant does feed them sugars. Certainly they're giving off CO2 as part of their physiology, but not to the same level. They can do this all at just normal atmosphere. You don't have to raise the atmosphere like what we do. Normal temperatures, and the bacteria is the catalyst. And so the idea that the plant roots have those nodules. And so here the picture is showing how the plant is feeding the bacteria sugar, and then the plant or the bacteria takes in the dinitrogen from the atmosphere and converts that to ammonia. So yes.
Why not? Why not?
Exactly. Yeah. And that's exactly, so we'll talk about this idea because if you can, well, they're actually doing kind of two big approaches. They're saying, okay, with these fields that we're trying to grow crops in, we want lots of nitrogen in there to satisfy the plant's needs. Can we, like, they're looking at, can we spike in a bunch of nitrogen fixing bacteria? And they've had good success with that. They've looked at the idea that there's plenty of bacteria in the soil that can fix nitrogen, but because that field has been fed nitrogen by humans over and over and over, those bacteria just kind of stop. They just kind of go to sleep with their nitrogen fixation. It's kind of interesting. It's like they're saying, well, I don't need to fix nitrogen because there's plenty of nitrogen here, so I'm just not going to do it. So now they're trying to wake those bacteria up, so to speak. So there's an effort with that too. Like they know, like, okay, this field has plenty of nitrogen fixing bacteria, they're just not doing it. So now we have to create a situation where we do it. Plus spiking in nitrogen fixers, plus the other idea of, can we grow plants in this field that have bacteria with them fixing nitrogen, and then can we plow that under and release, and then the bacteria release a lot of that nitrogen. So there's lots of strategies that they're looking at rather than, pumping ammonia gas into the soil from the Haber-Bosch process. So here we're looking at some nodules, and I put it in big bold letters, prokaryotic only. So even though I love mycorrhizal fungi and I think they're absolutely fantastic, fungi cannot do this because fungi are eukaryotic. We have yet to discover any eukaryotic organism that can do this. So far, it's only prokaryotic. I don't think that'll ever change. Only bacteria. And so here we're looking at some roots that have these nodules. So if you crack open those nodules, that's where the bacteria are hanging out. And so when the bacteria and the plant start communicating with each other, the bacteria release certain chemicals back to the plant that actually stimulates the plant to grow these nodules. And it's like growing a home for the bacteria, because then the bacteria move in, settle in, and they start doing their reaction. And so the bacteria have this dinitrogenase enzyme. And like I said, only bacteria have this enzyme. We've not discovered it in any other organism anywhere. And so what they're doing is they're taking that enzyme. Dinitrogen has a triple bond between the two nitrogens. And so that enzyme can break that triple bond. That's phenomenal. Triple bonds are really hard to break. And so this enzyme is incredible for its ability to do that. And only bacteria have it. And so what we see is that these nodules, so it's a really complicated process because the plant has to send out signals from its root into the soil, kind of doing a call out to, hey, are there any nitrogen fixers in the area? If so, follow my chemical back to me. And so then the bacteria pick up on that chemical and they start working up the concentration gradient to the root. But then, okay, so now the bacteria is right there. Now the bacteria has to convince the plant, I am safe to partner up with. I'm not an enemy. Let's get going. And so then the bacteria communicates back with a chemical to the plant, which the plant can understand. And then the plant goes, oh, all right, so let's get into this. And then the bacteria can gain entrance into the root tissue. They never get into the actual cytoplasm of the cells. They don't do that. That's too much of a boundary breach, and the plant would say, you're trying to attack me. So there's a boundary that you can't push beyond. And so it's the cell membrane or the membrane of the plant. And so the plant always keeps the bacteria surrounded by its own membrane, so it's not swimming, the bacteria can't swim around in the plant cytoplasm. And so they often refer to this as a controlled infection. The plant is controlling how far the bacteria are allowed. Like, respect my boundaries, only go so far, but I'll let you come into these nodules that I'm making for you if you set up your little home here and start working on this reaction where you can take the bacteria and create ammonia. And then that quickly gets converted to ammonium, H4+. So when this was discovered, it was like huge news in the plant world. And they named the type of bacteria that can do this Rhizobia. That's just a general term that they give. It's not a genus name or anything. However, they've taken some genus names and play on words off of them. So a genus that's called Rhizobium, and there's another one called Bradyrhizobium, Ezorhizobium. There's many others. These are just some examples. So if you see Rhizobium in the genus part of the word, you should think about, oh, they're Rhizobia. They're probably fixing nitrogen for some plant out there. And again, they're hanging out in those nodules. So the lower left picture is a kind of close up of some nodules on a tree, or I'm sorry, a plant root. And so here you can see, I think they kind of look pinkish to my eye anyways. And then the far right picture, I don't know if you can see a little kind of pinkish dots. This middle picture you can't see quite so well, but those pink, that actually has meaning. If I have time, I'll mention a little bit more about that. So they've discovered that legume type plants form this relationship with nitrogen-fixing bacteria. And so, listing out some of these legumes, you can see that those are really important commercial crop food production plants. And so having this partnership really helps these plants get access to nitrogen. So theoretically, you really shouldn't need to keep applying nitrogen to your field if you have these plants with the bacteria associated and they're doing their job correctly. So we'll come back to legumes, but well, let me look here at this table. So here you're looking at some of these different plants. And so you can see there's a range of different ones, even in non-legume phases for this here. And what did not make a list here is there's actually some conifers that hang out with nitrogen fixing, and they do it in their leaf tissue, not their root tissue. So sometimes you'll see nodules on leaves of needles on conifers. But anyways, so they're kind of focusing on mostly commercial produced things. But there's no aquatic one. Okay, so the rhizobial symbionts, those are the bacteria, but there's also free living bacteria that don't hang out with plants, that just fix nitrogen kind of for their own use. So be aware that there's free living ones too that could be in the soil, but we're kind of zooming in the ones that are out. That's a pretty special relationship. So let me go back. So these nodules that look pinkish, what's going on there is okay, very briefly, this dinitrogenase enzyme, there's one drawback to this enzyme. It's sensitive to oxygen. And the bacteria that have it most of them use oxygen. Not all, but many of them have to use oxygen for their own physiology. But this enzyme is sensitive to oxygen. So when the plant and the bacteria get together and they start doing this, sometimes you'll see this red tinge to the nodules. That's actually a molecule that's being produced by the plant. That's called, I'll spell it out here, leg hemoglobin. Leg hemoglobin. So if you take off leg hemoglobin, we have hemoglobin. It's part of our red blood cells. It's what makes our red blood cells red. And so these nodules look pink or red because of the plant version of hemoglobin. And so it's weird. What this leg hemoglobin does is it binds oxygen, similar to what our red blood cells do. Okay, it binds oxygen, and then it does a slow and controlled release of this oxygen to the bacteria. It's supposed to say release, slow controlled release of the oxygen. So you can imagine this leg hemoglobin, it latches onto the oxygen So now the dinitrogenase enzyme doesn't have free oxygen around it. So now the enzyme's safe, but the bacteria would suffocate because they don't get access to oxygen. So what this live hemoglobin does, it feeds out the oxygen to the bacteria. The bacteria use it right away. Then the live hemoglobin does that again. Bacteria uses it right away. And so you always keep the free oxygen low around this enzyme. So if you have this live hemoglobin working correctly and kind of feeding out the oxygen correctly to the bacteria, then this dinitrogenase enzyme survives, doesn't fall apart, and it keeps fixing nitrogen. So it's a really amazing way to deal with this enzyme being so sensitive to oxygen. And that's why it looks pink. The nodules, they look pink. So if any of you have tried I think it's the Impossible burgers, like the Impossible Whopper at Burger King, and they sell up their fake meat patties in the stores now too. I think it's the Impossible Company. Is that the name of the company? If you've eaten one of those plant-based burgers, you've eaten like hemoglobin. So I went to a plant conference where this company, the Impossible Company, I think, they gave a talk on how they came up with the Impossible Burger. And the trick is it's really hard to find plant-based products that taste like meat. And so they did a ton of research and then a ton of human, like sensory taste buds and your smell and all that. And they found the thing that was the most difficult to reproduce is the kind of that metallic taste of blood in meat. So when you eat, even if you cook meat well done, you still have that metallic blood taste and that you taste and smell. And that's what convinces you you're eating meat. And so like hemoglobin is like hemoglobin, like blood, red blood cell. And so when that gets mixed in, you are tricked into thinking that it's meat because it'll taste like that. I thought that was pretty ingenious. And so that's why they point to how successful their burgers are, that they taste so much like meat. So pretty ingenious. So, and using these nitrogen fixing bacteria and the nodules and all that is pretty cool. Okay, so nitrogen, that's a big one. Let's look at some others. So here's phosphorus and phosphorus. So here the images here are showing anthocyanin accumulation, where you have those purpley pigments showing up on the leaves in deficiencies. So phosphorus is hugely important for any organism because it's part of ATP and phospholipids, DNA, RNA. I mean, come on, it's huge. And so it usually is the second, they say the second most limiting element. Sometimes it can be the first because it depends on if in agricultural systems, sometimes you have enough nitrogen, but you still have issues with phosphorus. And so the reason I say that is that a lot of phosphorus in the soil is not readily available to the plant. It might be part of organic matter still, and so the plant can't break that down. It has to wait for bacteria and fungi to do it first. A lot of phosphorus, the state that it is in, is not soluble in the soil system. It'll be insoluble or precipitated out. And again, the plant can't get ready access to that by itself. You have to be careful with your pH because if you veer too far below 6 or if you go above 8 in the soil, phosphorus is going to be pretty impossible for the plant to get access to because in those pH conditions, the phosphorus precipitates out and the roots can't get access to things that are insoluble or precipitated. So phosphorus can be tricky. So even if you take a soil sample of your garden, send it off for analysis, they may say, oh yeah, you have phosphorus, but you can tell them to run what is my soluble phosphorus content. And then they will tell you specifically, yeah, you have a lot of phosphorus, but you don't have a lot of soluble phosphorus. And so you have to be reading your test results pretty carefully to make sure that you understand what's going on with your phosphorus. So deficiencies, it's kind of weird because when you see phosphorus deficiencies, the leaves actually look greener, dark green compared to normal leaves. It's kind of weird. And it's obvious. As long as you have good lighting, you can see that there's obviously a deeper, darker green. And then the purpley pigments can show up often. It's mobile, so that means the older leaves show symptoms first. If you keep the plant going and don't correct the situation, the plants look spindly is a good word. It just look kind of like a Charlie Brown Christmas tree. It's not very well developed, and it looks spindly. You really do have to make sure you know what your pH is, because maybe that's causing the problem. So that often is what needs to be addressed. And then sometimes you have to add in phosphorus if it's a true obvious deficiency. And then, of course, remember mycorrhizal fungi go a long way helping with allowing the plant to get access to, especially the insoluble or precipitated out, or the phosphorus that's in organic matter. Because remember, fungi, as well as bacteria, they're just really good at decomposing and recycling nutrients out of organic matter. All right, potassium. This one is also needed in very high quantities, NPK. So it's very, very soluble in the soil system. So it's not super common to have a deficiency in this because it's so easy to get into solution. It really depends. What's the big player here is how sandy is your soil? Because if you have sandy soil, potassium, yes, you can get deficiencies because the potassium will be in the water, and then because the sandy soil has such big pore spaces, it'll just keep percolating down with the water and go down into the groundwater, or the roots can't get access to it. Once the plant takes it up, it's super mobile through the plant body, so it can get into younger tissues no problem if there's a deficiency. And it's famous for this mottled chlorosis or mottled necrosis. Interesting. And the other idea too is that the tips and the margin, so that upper picture, I was trying to find more of the tip of the leaf or the margin of the leaf will go chlorotic and then necrotic. So look for that. If you're growing cereal grains like wheat or barley and so on, it can cause this terrible situation called lodging. Lodging of plants are when these cereal grains, the stem tissue gets really weak, and then, but they're busy producing grain, dry fruit essentially on the top of their stem tissue, and that causes the plants to flop over. So that's lodging, and the plants flop over, and then all the grains that you're trying to harvest are now on the top of the soil. You can't really harvest that with machines. It's really hard to use the same machine that you should be using. It can't work. And then, of course, you have cereal grains on soil where fungi are going to go, ooh, yummy food. And so they start attacking the cereal grains. So it's a real problem. You do not want this going on in your field. So luckily, though, potassium, because it's so soluble in the soil system, a good dose of it into your fields usually does the trick, as long as you don't have too sandy of a soil. And so potash, sometimes you'll hear them say, oh, I find potash. That's the version or the type of potassium that they usually act in. Okay. Calcium is another one that has been a kind of a surprising element study, because initially they didn't think it would do a whole lot, like maybe a few key things, but now they're finding calcium plays a pretty important role in the plant, more than what they thought. Luckily, it's pretty abundant in most soils. It's often not too bad of a problem. And they knew it was involved in cell division. So that means growth, because you gotta have cells dividing, so, plant growth. But this second messenger idea, it's heavily involved in a lot of physiological pathways, and getting a second messenger is a chemical in the plant that helps take a signal the plant is sensing from its environment and get it into the cells, get that message heard, so to speak, to the level of the DNA, so that the DNA can shift its gene expression. And second messengers help with that process. They're like intermediates in this process. And so they really help with the idea of taking some external signal and helping the plant make sense of it and then respond appropriately. And they didn't realize calcium would be so important for that. And it's showing up time and time again. Super important. Cell adhesion just means it allows neighboring cells to stick together really well. And so it just creates a nice coherent kind of the pectin in between the cells. The calcium is part of that pectin and really helps with cell adhesion. It's usually super obvious when there's a deficiency in calcium because the apical meristem region, so the stem tip, gets deformed and gets hit hard. And so the young leaves that start to grow look all twisted and deformed. They don't fully leaf out and flatten their blades, so they're all kind of crinkly and puckered looking. It's very obvious when you see it with the naked eye. If you grow plants hydroponically and have a calcium deficiency, if you lift up the plant out of the solution, you'll see that the root growth is terrible. And that's a dead giveaway that you've got some calcium issues. If you're growing tomato in your garden, I've had this happen more than once. So that lower picture is actually green tomatoes on the vine still on the plant. And now that black is the bottom of the tomato fruit that's forming. That's called blossom end rot. It's horrible to see that in your tomato plant. Luckily, it is, especially, if you've ever grown tomatoes, you know that they kind of successful or grow tomatoes in succession. So it's not like they make all their tomato fruit all at once. So if you see this happening with some of your early fruit, spike in calcium, and then the remaining fruit that's starting to form, it should be okay. You should be able to salvage what's left in terms of that tomato plant growing more fruit. But I've had this happen more than once. It's so disappointing. Lettuce gets the tip of the leaves gets obvious burn look to it, that necrotic idea. And poinsettias, if you grow poinsettias for the holiday season, that can really mess with the shape of the bracts. People say it's the flower, but it's really a modified leaf that's brightly red. And then once it gets into the plant, it's immobile. Okay, we'll continue on with a couple others on Monday. And then we'll get into the next topic as well, which is looking at just how to manage your soil for growing plants.